Welcome to the AAPS Immunogenicity 101 course module on humanized mouse models. This material is presented to you by Dr. Jack Ragheb, Chief Medical Research Officer of the Laboratory of Immunology, CDER, US FDA, and Dr. Teresa Goletz, Director of Medical Sciences and Clinical Immunology at Amgen. The objectives of this course module are shown here. The speakers will cover basic tools for predicting immunogenicity. They'll give an overview of the various humanized mouse models, describe the applications of these models, as well as detail the current limitations and potential solutions. Therapeutic proteins have the potential to elicit an immune response in the receiving host, and many factors, both intrinsic and extrinsic to the molecule, can influence immunogenicity. More information on these can be found in other modules. In order to better understand the immunogenic potential of a therapeutic protein, multiple tools have been developed or adapted to predict or assess immunogenic risk. These tools can be applied at various stages of drug development. This slide shows three main approaches used to describe tools used for immunogenicity prediction. The first category are in silico tools. These refer to computer-based algorithms and models which assess a particular attribute of a therapeutic protein. Some of the most frequently used tools identify peptides within a primary protein sequence, which have a high probability of binding to major histocompatibility, or in humans, the human leukocyte antigen class II molecules. The identification of peptide HLA class II binding hotspots serve as an indicator of T helper cell recognition, a critical step for T cell dependent B cell responses, or the development of anti-drug antibodies. Such hotspot analysis has been reported to correlate with clinical immunogenicity. The most common application of this type of assessment is in early drug development and helps to better inform lead candidate molecule selection, or in the case of engineered sequences, these tools enable the assessment of the potential creation of neoepitopes which may be recognized as foreign or non-self to a host. The second category is in vitro tools, and these refer to a variety of in vitro cell-based and non-cell-based assays. The most frequently used approach is cell-based immune activation assays. There are many varieties of these assays and many types of readouts. I'll provide more details in the following slides. These assays are often applied in a comparative manner, for example, in assessing the impact of quality attributes, such as protein aggregation. These same assays can also be used for immune monitoring for cases where the mechanism of action of a molecule might alter immune function. Finally, the last category includes in vivo models, which is the topic of this module. These models, which include transgenic or humanized mice, have also been used in comparative assessments. The most frequent example is in the comparison of the biological or immunological impact of quality attributes, such as protein aggregation, and differences in post-translational modification or other characteristics.